Shri Gurubhyo Namaha children, I am happy to welcome you all for our online teaching academy XII. Today we are going to discuss about the fourth chapter, Principles and Processing of Biotechnology. In the previous class we discussed about the steps in rDNA technology, that is recombinant DNA technology. Now we are going to see the tools for biogenetic engineering. Genetic engineering is also called as rDNA technology. R represents recombinant DNA technology or gene cloning. In order to generate recombinant DNA molecule, that is rDNA molecule, certain basic tools are necessary for the process. The basic tools are enzymes, vectors and host organisms. These are the three important tools, basic tools for constructing or building rDNA, that is recombinant DNA. The most important enzymes required for genetic engineering are, the first one is restriction enzymes. Children, please follow me. Restriction enzymes. Second one is DNAligase, and third one is Alkalinephosphatase. So these three enzymes are very important enzymes which is required for genetic engineering. Now we will see the first enzyme, restriction enzyme. The two enzymes responsible for restricting the growth of bacteriophage in E. coli, that is Escherichia coli, were isolated in the year 1963. One was the enzyme which added methyl group to DNA, and another one is cut the DNA sequence, DNA fragments. So the later one, the cut the DNA fragments, is called restriction endonuclease enzyme. This restriction endonuclease enzyme is used to cut the DNA sequence or DNA fragments. Okay. So another enzyme is very essential to add the methyl group to the DNA. So there are two enzymes, one is adding the methyl group into the DNA and another one is cut the DNA. For cutting the DNA sequence, the name of the enzyme is restriction endonuclease. Clear? The restriction enzyme is an enzyme that cleaves DNA into small fragments. You know very well DNA is a double strand. One strand running from 3' to 5' end and another one is running from 5' to 3' end. So with the help of restriction endonuclease enzyme, we can cut the DNA sequence into small fragments. So restriction enzyme is nothing but it is an enzyme that cleaves DNA into fragments at or near the specific recognition site, very important word, that is specific recognition site within the molecule known as restriction sites. In each DNA sequence they have one important site that is called restriction site, that is the recognition sites. Clear? There only DNA enzyme, that particular restriction enzyme cut the DNA into small fragments. Clear? Based on the mode of action, the restriction enzymes are grouped into or classified into two. One is exonuclease and another one is endonuclease. The name itself you can easily understand the concept. Exo means outside, it acts outside. Endonuclease means it cleaves the DNA fragments into small pieces. It breaks the internal phosphodiester bond, that is called endo. Endo means inside. Can you get my point, children? Yes. First one, exonucleases. Exonucleases are one in which remove the nucleotide at a time from the end of a DNA molecule. You know very well, DNA is made up of millions and millions of nucleotide sequence. This exonuclease remove the nucleotide at a time from the end of DNA molecule. Example for exonuclease is BAL31, exonuclease 3. These two are the examples of exonuclease. Okay, and then next one is endonuclease. So endonucleases are the enzyme in which it breaks the internal phosphodiester bond within a DNA molecule, within a, that means endo. Example, Ind3, EcoR1, BAMH1, TAC1. These are all the examples of endonucleases. Are you get my point, children? Shall we move on to the next slide? Yes. See the tabular column here. These are some of the examples of restriction enzymes, and then their recognition sequence, and then fragments. The first restriction enzyme is ALU1. It is obtained from the microbial source Arthrobacter luteus. This particular enzyme recognizes the sequence AGCT and then complementary base TCGA. It cut the sequence, it cut the fragment into two equal parts. That is, the end is called blend end. It is, the cutting is symmetrical. It cuts, it divides the fragment into two equal parts, two equal sequence. The type of end is called blend end. One more restriction enzyme is there, that is called BAMHI. This is the name of the restriction enzyme. It cut the DNA sequence like this. This end is called sticky end, zigzag cutting. Okay. The next one is EcoR1. EcoR1 is nothing but Escherichia coli. It is obtained from the microbes, bacteria, E. coli, Escherichia coli. The name of the enzyme is EcoR1. It also cuts the fragment into like this, zigzag, sticky end. Clear, children? There are three main classes of restriction endonucleases. They are type 1, type 2 and type 3. All these types, type 1, type 2 and type 3, they are different, slightly different in their mode of action. All the three are not same. They have very slight difference in their mode of action. Among the three, type 2 restriction endonuclease, this type 2 enzyme is preferred for use in rDNA technology, as they recognize and cut DNA sequence within a specific sequence, typically consisting of 4 to 8 base pair. So among the three type 2 is very important. This type 2 enzyme, restriction endonuclease enzyme, is preferred for rDNA technology as to recognize and cut the DNA within a sequence typically consisting of 4 to 8 base pairs. So this particular enzyme recognize the sequence of 4 to 8 base pair and cut the DNA sequence into small fragments. Next, one more thing, the restriction enzyme Ind2 always cut the molecule at a particular point, recognizing a specific sequence of 6 base pair. So this particular enzyme cleaves, divides, cut the sequence having 6 base pairs. This sequence is known as recognition sequence. Please listen, this is called recognition sequence. Restriction endonucleases are named by standard procedure. They have some procedure. We already discussed about what are the restriction endonuclease enzyme. For example, Ind2, EcoR1, like that, right? So for naming the enzyme, they follow some of the procedures, standard procedures. The first letter of the enzyme, the first letter of the restriction enzyme indicates the genus name followed by the two letters of the species. Can you get my point, children? Yes. While naming the enzyme, we have to follow some standard procedure. The first letter should be the genus name followed by the two letters or species name. Then comes to the strain of organisms, finally a Roman number, numerical number indicating the order of discovery. For example, I am taking here EcoR1. EcoR1 is the name of the restriction endonuclease enzyme. Is it clear, children? Yes. How it pronounced? EcoR1, right? EcoR1. Here E represents a name of the genus Escherichia. See here. And then CO. CO is come from coli, the species name Eco. After then strain RY13. Here R is a strain, and first endonuclease to be discovered, so that they put the name 1. So EcoR1. E is the genus name, CO is the species name, R is the strain, and 1 is the discovery, order of discovery. Can you get my point? Yes. It consists of two different antibiotic resistant genes and recognition site for several restriction enzymes. This sequence is referred to as restriction site, and it is generally palindromic, which means that sequence in both the DNA strands at this site read the same in 5' to 3' direction as well as in 3' to 5' direction. For example, read this word. What is this? Malayalam. What is this? Malayalam. How many letters are there? 1, 2, 3, 4, 5, 6, 7, 8, 9, right? From here just read M-A-L-A-Y-A-L-A-M. From left to right the same M-A-L-A-Y-A-L-A-M. From right to left the same letters are there. No? Such type of sequence is called palindromic sequence. Clear? So this Malayalam, this phase is read the same in the either of the direction. This phase, this word can be read the same thing. You can read this in the same either direction as it is. So M-A-L-A-Y-A-M is both in left side as well as in the right side. So palindromic repeats a symmetrical repeated sequence in DNA strand. Symmetrical, that means two strands have the same one. See here, one strand starts from 5' to 3' end, and another strand starts from 3' to 5' end. These two are the strands of DNA having the base nucleotide C, A, T, T, A, T, A, T, A, T, G. While reading this G here, T. So complementary base pairs are present. That is called palindromic repeats. So C complementary base G, A, T, T, A, T, A, A, T, A, T, A, like that. The complementary bases are there. So one strand runs from 5' to 3' end, and another one runs from 3' to 5' end. Both these strands have the complementary bases. While reading from left to right, G A T T A T A T A T G, like that. That is the sequence. Okay. C represents cytosine. Then A represents adenine. T represents thymine. What is this? Adenine, guanine, cytosine, all these are bases. Okay, nitrogenous bases. So this complementary sequence, complementary base, G always base pair with C. C always base pair with G. These are the complementary sequence. So, from left to right CATTA, the same sequence is there in right to left. Is it right? From left to right, this direction 5 dash to 3 direction, the same sequence is there in 5 dash to 3 dash in another stand. Such type of repeats is said to be palindromic repeats. Very important 2 more question. What is palindromic repeats? A symmetrical repeated sequence in DNA strand, that is called palindromic sequence. The exact kind of cleavage produced by restriction enzyme is important in the design of rDNA technology. So, already you know that restriction enzymes have the ability to cut the DNA fragments. It cleaves the DNA fragment, right? So, they have some property, cleavage property. Some cleave the both strand of DNA. Some means some enzyme cleave the both strand of DNA through center, resulting in the blunt end. See the sequence here, GAATTC. This is a one strand, starts from 5 dash to 3 dash end. Another one is 3 dash to 5 dash end. So, with the help of EcoR1 enzyme, this enzyme cut the sequence in the middle. So, it cut the sequence through the center. Now, we are getting symmetrical cuts. See here, symmetrical cuts. But EcoR1 cut the sequence into a dissimilar, that is asymmetrical. This type of cut is called sticky end or cohesive end. I am repeating once again. Some cleave both strand of DNA through the center, resulting in the blunt or flush end. They are known as symmetric cuts. But some enzymes cut in a way producing protruding and recessed ends through a sticky or cohesive end. Such cuts are called staggered or asymmetrical cuts. So, EcoR1 has the ability to cut the sequence into dissimilar shape, that is sticky, asymmetrical. But SMAL, that is the name of the restriction enzyme, cleave the sequence into two equal. It cleaves through the center. Now, we are getting blunt end. Okay, blunt means symmetrical cut. Are you get my point, children? The other two enzymes, we discussed the restriction enzyme. The other two enzymes, DNAligase and alkalinephosphatase, is also very essential in rDNA technology. See here, the other two enzymes that plays an important role in rDNA technology are, what are they? DNAligase, and another one is alkalinephosphatase. Shall we move on to the next one? DNAligase? Yes. DNAligase. In short, it is called as what is this, children? A nick sealing enzyme. Very good. Nick sealing. That particular enzyme is very essential in rDNA technology. Why? With the help of restriction enzymes, all the DNA fragments are broken. So, with the help of DNAligase, this enzyme joins the fragments, join the cutted fragments. So, it is called as nick sealing enzyme, joining enzyme. Clear? Yes. DNAligase enzyme joins the sugar and phosphate molecules of double stranded DNA with 5' phosphate and 3' OH end in an adenosine triphosphate dependent reaction, ATP dependent reaction. This is isolated from T4 phase. So, DNAligase enzyme is isolated from T4 phase. See the picture here, children. What is this? This is a DNA, one DNA, and this is another DNA. Is it right? So with the help of this DNAligase enzyme joins. So this is a one DNA, this is also a same DNA. It joins the sugar and the phosphate molecule. Here 5-phosphate. Here this is 5-phosphate. PO4. 5-phosphate and the 3-OH. So DNAligase joins the 5-phosphate and 3-OH in an ATP dependent reaction. Now these two strands of the DNAs are joined to form a long linear structure. The next enzyme is alkalinephosphatase. It is a DNA modifying enzymes. It has the ability to modify the sequence, not at all sequence, the end of DNA sequence. Okay, so it is a DNA modifying enzymes. It can add or remove specific phosphate group at 5' terminals of double stranded DNA or single stranded DNA or single stranded RNA. Thus, it prevents self-ligation. Okay, this enzyme is obtained from bacteria and calf intestine. See the picture here, children. This is the DNA sequence. The plasmid cut open. These plasmids are cut open. With the help of DNAligase, we are getting the plasmid self-ligation, recircularization of plasmid. We can remove the phosphate here from here. This alkalinephosphatase enzyme removes the phosphate. Remove the phosphate. Now we are getting OH on both strand and OH on both strand. So there is no ligation takes place. DNAligase will not act on this. DNAligase only act on 5-phosphate end with 3-OH end. So DNAligase only joins the phosphate end with the OH end. It will not act on OH. Okay. So this alkalinephosphatase removes the phosphate on both strand. Now the both strand having OH. So then foreign DNA fragment having two strand, the same two strand. One starts from 5' to 3' that are having a 5-phosphate and another one having a 3-OH. Now with the help of DNAligase joined this and we are getting finally the rDNA. So this enzyme alkalinephosphatase is also essential in rDNA technology by add or remove some specific phosphate group at 5' terminal of double stranded DNA or single stranded DNA or single stranded RNA. Clear, children? Thank you, children. We will see in the next class.